Good morning to everyone, and a warm welcome from a warm Worcester in the Western Cape, South Africa. The Parrot Breeders Association of Southern Africa would like to welcome you to our first webinar for 2025. And we've chosen a very interesting subject to start off this year, and there'll be quite a few interesting ones to follow. So please keep your eyes out for what we're going to be presenting next. We've chosen a subject that is very, very important for our industry, and we've asked for a lot of participation, and we have had massive anticipation so far, and we envisage a very exciting webinar. But please bear in mind that we are limited with time, and we will probably not address all the aspects today, but we will definitely be looking at follow-ups and to try and get through the subject in a couple of webinars to follow still. We would like to first thank our associates, our sponsors, and our advertisers, and you'll see some clips of them every now and then, and in my background. It's important that we can't do something like this without their participation. They make these things possible. And this is a truly interactive platform that we're using, and we would like the people to comment. You can either comment on the Facebook or on the YouTube links, and let us know where you are from. We've got Gert, welcome Gert from Heidelberg. For practical reasons, someone from Uppington. We are going to be doing the whole presentation in English because we have a lot of international guests that have tuned in as well. So please bear with us. For those that want to put a comment or a question in Afrikaans, they're more than welcome. The important thing is that it's interactive and we want your participation. Our panel will be looking at questions and comments that are posted, and we will be going through them. Obviously, with time constraints, we won't be able to address every single question directly, but we will take note of them and we will try and get answers for them, and they will be published on some other forum in future to answer your needs. So please, we will take note of every single comment and question. It's just that we have to get the program nice and flowing. Let me just see what I might have forgotten here. The format that we'll be following is we've divided the presentation or the webinar into 4 sections. The first section, we will be looking at the virus. The second section, and the research that's being done up until present. The second section will be on the laboratories and the testing and the different tests available, testing techniques, and how to interpret these results. The third section will be the way forward. And this is the challenging part because it doesn't help we discuss something of the past and we don't have a route forward. So that's going to be very nice and interesting. And then the fourth section is a short question and answer session. We'll probably only have time for 4 or 5 questions, but please submit them and we will address as many as we can. We've condensed a lot of questions for our panel so that they know what aspects to address. And then, with any further ado, I think we can start by introducing our panel. And we are very thankful for the time they've put aside to put effort into this webinar. And I really hope they find something of value out of it as well. And I definitely know that all our guests definitely will. So the first panel member we would like to introduce is Prof. Robert Bragg.
I'm Emeritus Professor Robert Bragg. I'm currently retired, but I'm still affiliated with the University of the Free State. When I was working at the University of the Free State on beak and feather disease virus, my research group was primarily looking at vaccine development and improved diagnostics. My current research is on resistance to disinfectants.
Welcome, Prof. Bragg.
Thank you.
Our next participant will be Dr. Anton van Zyl.
Hello, I'm Dr. Anton van Zyl from the University of Cape Town. I'm currently associated with the Centre for Bioprocess Engineering Research and also the Biofarming Research Unit, which is where all the BFDV magic happens. Now, I started my career at the University of the Free State under the supervision of Professor Rob Bragg, and we focused on the development of diagnostic assays for BFDV. Since then, I've worked on blue tongue virus, HPV, and some other emerging diseases. But in recent years, I've found my way back into BFDV research, specifically focusing on the development of vaccines. Now, this work happens at the Biofarming Research Unit under the supervision and guidance of Professor Ed Rybicki and Professor Inge Hitzeros. Now, our work specifically is about making vaccines in plants. This is a hugely exciting field, and I look forward to discussing it today.
Good morning.
Welcome, Alta.
Thank you.
Our third scientist involved in the research is Dr. Dirk Verwoerd. Dirk has got a BSc Agric, his BSc with honors in poultry diseases, an MSc in animal-human ecosystem health. He's got 35 years of experience in the veterinary field, mixed between practice, research, and academia. He's also had a stint in the pharmaceutical industry, and he's involved with intensive animal production industries, and he's representing Parrot Breeders Solutions today. Welcome, Dirk. Dirk, can you hear us?
I've got a few sound issues from, I don't hear you very well. I hope you can hear me, but
Thanks, Rob.
I gather you've introduced me. Thank you very much for the opportunity.
Thanks, Dirk. Our next panel members are people that are involved with our laboratories, and we are very thankful for their participation. Our first participant will be Dr. Dennis York. Dr. Dennis York has been involved with molecular diagnostic services for many, many years. And as you can see there, he's done 32 publications already, a very experienced scientist in this field. Morning, Dr. York.
Morning, thanks very much.
Our next participant will be from SMT, Christine Stradom. Can we?
Good morning, everybody.
Okay.
Just a small introduction to me. I am Christine Stradom, and I'm a veterinary technologist with a master's degree in veterinary science. I obtained it from the University of Pretoria. I have over 12 years worth of experience in the poultry industry. For about half of that, I have also been in the parrot industry, mainly with the avian influenza and Newcastle testing. And I'm currently quality manager of SMT Vet. I started at SMT Vet in 2021, so I've been there for about 4 years right now. My skills in diagnostics range anywhere from virus isolation, serology, microbiology to molecular biology. My preference in the industry is the study of viruses, and I have been published alongside names such as Prof. Celia Polning, Dr. Laura Roberts, and Ms. Diani Ralph. I have knowledge in the validation of new methods and verification of methods according to the WOAH standard. And in my free time, I like reading, walking, and spending time with my friends and family. That's a little bit about me. I hope you enjoy the webinar. Thank you.
Welcome, Christine.
Hi, thank you.
Right, and our last but not least panelist is Daria de Villiers. He's representing Lumigen. Daria has a BSc in biochemistry, also with the honors in biochemistry, life science and physical science, MSc in biochemistry, and an MBA. He is also head of innovation and commercialization of Lumigen Laboratories. Welcome, Daria.
Morning, everyone. We're excited.
Right, as I think you can see, we have a very, very experienced panel here today, and we hope to get just a fraction of their knowledge. It'll help us a lot along the way. So please participate. We'll try and keep it flowing, but we would like to see your participation. Here we have greetings from someone from Plet area. We haven't seen any of our international guests yet. We hope to see them coming up soon. We'll start off with a little introduction video about a bit of the background on citizen beak and feather disease virus, and from there we will start with our discussions.
Welcome to this online webinar about Psittacine beak and feather disease virus. This is a disease that's devastating the parrot industries worldwide. The objective of this webinar is to gain open and frank discussions around various aspects related to the disease. Firstly, we need to get better understanding of the virus that's causing the disease as well as the clinical signs. We also need to look carefully at the diagnostic procedures, particularly whether the PCR test is reliable for detecting asymptomatic carrier birds. Is there a need for serological tests? Then we need to look at control options. Because the virus cannot be cultured, the conventional methods of making vaccines are not available. So the only options for vaccines is molecular-based vaccines. Some discussion is needed around this. Another treatment option would be the development of antibodies. These could either be monoclonal antibodies or recombinant antibodies. And finally, and most important, would be biosecurity and disinfection. Because Psittacine beak and feather disease virus is a non-enveloped virus, a naked virus, it is extremely resistant and can survive for weeks or months in the environment. So good biosecurity, good disinfection, good use of quarantine is essential for the control of this disease. I hope this webinar is going to be fruitful for everybody, and I look forward to taking part in it. Thank you very much.
Thank you, Prof. Bragg. I think that's given us a bit of an idea of what we're dealing with. I'm going to give each of our other two scientists opportunity to add a bit of a comment if they have something they would like to add to the introduction, and then we'll start getting into a bit more specifics. Dr. Alta, can we get you up to add a bit if you like?
Yes, so like Prof. Bragg said, not being able to cultivate the virus has had a huge impact on vaccine development. So here at UCT, we've specifically focused on developing vaccines, but of a recombinant nature. And we've looked at different types of vaccines, which include a DNA vaccine, a protein-based vaccine, which is just made up of that outside capsid of the virus. And then most recently, we looked at an mRNA vaccine candidate as well. And to keep our mRNA safe from degradation, we encapsulated it in a plant virus. So that's all been very exciting research we've done. We've managed to test this in African grey chicks together with Dirk. And studies were really successful, I think. It laid a good foundation for what we need to do further. And we did show that both the mRNA and the subunit vaccine induced really nice antibody titers in the birds. But, and this is the big but, because we cannot cultivate the virus, it's really difficult to test whether these antibodies actually do their job at stopping the virus from entering into the cells of the birds. So there's quite a bit of research we still need to do here. And we also need to do, the first study was just sort of a pilot study. Do these vaccines even work? And, doing it in African grey parrots, that was really hard. We haven't been able to do that for years. And then we got connected with Dirk. So we're really grateful for that. So going forward, it would be optimizing our vaccine schedule. Can we combine these two vaccines in different doses? So vaccinate first with a subunit and boost with an mRNA vaccine or vice versa, things like that. And then we really need to think about how we're going to test efficacy of these vaccines.
Thank you very much, Dr. Van Zyl. Here we have a guest all the way from New South Wales in Australia. Welcome to Jody and Nathan. We're glad you could join us. It's really nice to see some international participation. Right. Our next input, we'll ask Dr. Dirk Verwoerd to add something if he has something we'd like to add to the background that we've just received. Dirk, you up?
Okay, can we upload this presentation?
I think let's just give a quick background. The slides have got a lot of data on it, and that makes it very difficult for people to read it quickly. Just give us a bit of additional information that you'd like to add to the introduction.
Yeah, so Skalk van Wyk and myself, we form the Parrot Breeding Solutions Group, and we've partnered with several eminent other research groups, most notably the UCT group, most recently, very exciting stuff there. Tested a number of prototypes. We've taken the position that we will only do this work in African grey parrots. We're really interested in finding a solution for the African situation. Possibly in the future that is applicable, could be applicable to also endangered Cape parrots, but African greys are really the focus. So that's our experimental model, which makes it very expensive, but it's still, I think, appropriate to what we are dealing with here in this part of the world. So before that, we also partnered with Design Biologicals, and we have, together with them, developed a experimental capsid vaccine. And we've been testing it over the last few years in several hundred chicks, going forward, going backward, trying different concentrations, different adjuvants, different things. So really it's still at the experimental stage, and I'll go through it in a little bit more detail in the slides. But we're at the verge, I think, of breakthroughs with all these groups. And in future, of course, we envisage a number of tools in the toolbox. Rob has mentioned the importance of biosecurity and disinfection. That's absolutely crucial. Those are our most valuable tools at the moment. In our group, we focused very much on nutrition. I think the addition of vaccines as a preventative tool to a proper nutrition and diet, and giving the bird the immunostimulants, everything that's in there, that's also crucial. So if you want to, I can go through the presentation, just a few slides, and maybe give you some basis.
I think, Dirk, we'll flow into them as the discussion starts picking up. I'm just going to give Prof. Bragg opportunity to add something that he might have found after your comments, and then we can start with our discussions.
Okay, thank you very much. As Alta mentioned, she was working with Prof. Ed Rabitsky, and him and I, many years ago, decided to start looking at beak and feather, not because it's easy, but because it's difficult. And between the two of us, we did quite a few things. I started off asking through the Aves Zandam for birds, and we were inundated with sick birds. First of all, we tried, as Alta mentioned, that we can't cultivate the virus. So this takes out all the easy steps. If you could cultivate the virus, it would have been sorted out a long time ago. So first of all, we tried to cultivate the virus. That didn't work. Then with Alta's hard work, we looked at some of the serological tests, looking at the ability of the virus to hemagglutinate red blood cells. It's a very good test, serological test. And then after a visit to one of the biggest bird breeders in the world in the Philippines, it came to light that they were using commercial poultry ELISA tests to test in the parrots. And I was under the impression that this is not going to work. So when I got back, we set up an experiment where we vaccinated parrots with Newcastle disease virus. We did the conventional hemagglutination inhibition test, and we did the ELISA test. And we got very nice immune profiles with the hemagglutination inhibition test. But unfortunately, with using the anti-parrot, the anti-chicken, we got very mixed results. So we had to develop a competitive ELISA, which I think is one of the main things that Alta was involved in. Then we looked at a DNA vaccine, because at that stage, it looked very promising, because the concept was you vaccinate the individual once and you get lifelong protection. Now, for a long life parrot, that would be brilliant. So we set up a DNA vaccine experiment. The PhD student that did that is now the vice dean at UJ. So I've been able to produce some good quality students and a good few publications, but little else. We did produce the DNA vaccine, and we did vaccinate budgies, and we got an immune response. But as Alta also mentioned, testing the efficacy is extremely, extremely difficult. We also been working on a yeast-based expression system, and we have a patented yeast that we can use. We got some success with expressing von Willebrand's protein, which is a human blood clotting protein. And we decided to see if we could express the coat protein of beak and feather into the yeast. This we did. We were able to see the coat protein of the virus sitting on the surface of the yeast. But unfortunately, due to lack of funding, we didn't get much further with it. But I think one of the most interesting things that we did is we received a group of confiscated African greys that came in from, I think, from Senegal. That tested positive for beak and feather at the airport, and they were donated to us by Nature Conservation. And we kept these birds for a number of years. They were asymptomatic. In other words, they weren't showing clinical signs, but they tested positive on arrival. And we tested these birds consistently over years. And the bottom line is we were getting very variable PCR results. Sometimes a bird would test positive. Sometimes 2, 3, 4 times the bird would test negative, and then it would test positive again. So it's apparent that this virus goes into a carrier state somewhere in the bird, and if the virus isn't present in the blood, the PCR test doesn't work. So this begs the question, do we rather need to start looking at a reliable, good serological test like an ELISA, where we can check the exposure of whether the birds have been exposed to the virus and not if the virus is actually present. And then, of course, as mentioned, biosecurity, good quarantine remains essential. Remember, this virus is a naked virus, a non-enveloped virus, and it can remain in the environment for months. So unless you're having a good quality disinfectant that can kill the virus, inactivate the virus, you're going to have a consistent problem. And it's an extremely, extremely difficult virus to kill. If anybody tells you their product kills beak and feather, unfortunately, I must say they're not being perfectly honest with you, because as we've already mentioned, you can't grow the virus. So if you can't grow the virus, you can't test it. The best you can do is you can test whether the disinfectant works against other closely related viruses. Having said that, I think I've had my say. Thank you very much.
Thank you very much, Prof. Rob. I think 1 or 2 points that have come out of this is that it's impossible to culture the virus, and that our tests results can be very variable if we're only using the PCR, and that we need to develop separate some different testing techniques, and we'll get to that a bit later. But a pertinent point came up a while back by some prominent agriculturals that they suspect that the virus might have mutated or can possibly mutate. Dr. Alta, can we ask you to maybe comment on the possibility of the virus mutating? We know there are several substrains, but can you probably comment on that for us?
Well, I think it's not impossible. And, with the sequencing technologies these days, we really can do a really nice study also to see what happens over time, especially if you have, let's call it a cohort of birds that you can also study over time. But finding mutations, it's not always easy. But we already know that different geographical areas have different geographical lineages of the virus. And just in terms of geography, that might affect what the immunogenicity profile of that virus is going to be and whether the sequences are the same or not. We already know from work we did in our lab, like a coat protein gene from a Cape parrot is not equal to the coat protein gene from an African grey or a budgie. So we know that there are inherent differences with this virus, and that's also what makes it a little bit difficult, right? I mean, I can also go a little bit into saying we tried to develop a challenge material at UCT, ended up being more of a DNA vaccine candidate than a challenge material. So we made a fake virus. It looks exactly like the BFDV, but it's not infectious, really. We tried to test it in quail cells in the lab, but we know viruses are very specific, so we couldn't get infection profiles in these quail cells. And we don't have commercial parrot cells that we can play around with in the lab. And then again, together with the work we did with Dr. Verwoerd, we tried to use it as a DNA vaccine, but the titers were really low. So yeah, the virus, I'm sure it's mutating. We also did a study in hog's back on an on a Cape parrot population where we screened a lot of birds. We did the PCR test. And then a couple of years later, we went back and screened the birds again. Obviously, you don't know if it's the same bird necessarily, but we got such variable results that it almost looked like none of the birds had beak and feather by the time we test them again a couple of years later. So I do want to also agree with what Prof. Bragg says. It looks like the virus goes into some sort of a slumber state as well. And perhaps when a bird is immunocompromised or stressed in some way, shape, or form, it just goes rampant in the parrot again. So yes, mutations are real. That's something we need to consider with vaccine development, and whether different species of psittacines will have different vaccines because of these mutations. So we need to understand more about the fundamentals of what will give us a good immunological response. But again, it's a really difficult thing to do because we cannot cultivate the virus from these different species.
Thank you very much, Dr. Van Zyl. Getting back to some of the points that you brought out here is that the carrier state is one of our biggest challenges, and I think we all realize that. But once a bird has been infected, does it stay infected, or does it heal? Can it become immune to the virus, or does it stay infected and then under certain conditions does it flare up again? I see Prof. Bragg wants to make a comment. Also, can I give you a first option before I put it to the rest of the panel?
I think Dr. Verwoerd would be a good one to ask this because he's hands-on with the birds all the time. But I do think maybe there are two scenarios here, and we need to consider both. And that is that perhaps it just goes into a latent stage and it just hides under the surface. And perhaps in some instances, the birds are they just get better and they get rid of the infection. So I'm not really sure, but I think Dr. Verwoerd is the better one to answer that.
With that, can I ask that we quickly cut over to Dr. Verwoerd that he can add his comments, please? Dr. Verwoerd?
Anton, would you like me to carry on? Sorry, I've got a audio problem on this side. I do not hear anybody.
Okay.
So if you could just talk your and if you want me to go ahead, then I'll do so.
And I said we discussed the situation that there seems to be that the virus goes into a latent phase, and then we have a bird that is either a carrier, and we don't know if they are carriers, or some of them have healed from the disease. And Dr. Van Zyl was saying that you have some more experience of this potential carrier state. Would you like to elaborate a bit on that?
I'm sorry, you'll have to type your question, then I can respond. Okay, I see all the comments typed. I cannot hear anything.
Thank you, Dr. Verwoerd. We'll get back to you. We will send you the question, and I will, in the meantime, refer to Prof. Bragg.
Yeah, thanks. I don't really want to play devil's advocate here, but I think it's important that the points be raised. When we're talking about vaccines, they have to be registered. So you can't just make something in the lab and then sell it. It needs to be registered, and there's a big process going through the registration of vaccines. The other very important comment is all of these are GMO vaccines. So before you start using these vaccines experimentally or using them in the field, you have to get GMO approval for release. So this is, as I say, I'm putting a blanket on everything here, but we need to look at the legislation around the use of vaccines, and particularly GMO vaccines. Sorry, but it needs to be said.
Thanks very much, Prof. Would you like to comment on the fact that the virus goes latent? You mentioned it as well. How do we interpret a bird that has tested positive, later tests negative? Is he always a potential carrier, or can he develop immunity?
Look, the best example we have was with those African greys. They were wild-caught African greys that came in. They were asymptomatic. They were beautiful birds. but they tested positive at the airport and they were confiscated. And we kept those birds for some 6 or 7 years. And for months, the PCR would test negative. And then all of a sudden it would come out and we'd get a positive test again. So it's highly likely that the virus is hiding somewhere. We did look at some literature. There were some possibilities that it could be going into the bone marrow, or it could be going in. I think there was discussions about it could be going to the liver. But it became quite obvious from these birds that we were looking at that if they're non-symptomatic, if they're not showing clinical signs, the PCR test is pretty unreliable. The PCR test is brilliant for birds that are showing clinical signs. So if you got the virus in the blood, voila, the PCR works well. But if the virus isn't in the blood, the PCR comes up negative. So you're going to get inconsistent results, and the carrier state of the birds is a major issue. And this is, everybody rushes off and they go to the auctions and they buy their bird at the auction, and they've got huge collections of parrots, and they slap their new bird right in the middle of their parrot collection, and you're bringing in everything that new bird has got with it. Quarantine, spend your money on building a quarantine station for yourself. Put your birds in quarantine, leave them in quarantine for at least 6 months. Because remember, stress will also induce any carrier birds to allow the virus to come out. So you've got the stress of the bird going to the auction, you've got the stress of the bird moving to a new facility. That bird is stressed. So the likelihood that stuff's going to come out is high. But have it in quarantine. Don't put it in the middle of your collection and then all of a sudden you find it's got beak and feather or it's got something else. Your best investment is build a quarantine station for yourselves. Thanks.
Thanks very much, Prof. Bragg. Can we maybe cut back to Dr. Verwoerd? Dirk, the request from our technical team has been that maybe you can connect on your phone to here and then use your computer to give us sound you're talking through. Will you try that? You must please unmute yourself, please, there, Dr. Verwoerd. Dirk, can you please unmute yourself? Okay, I think let's, while we try to connect with Dr. Verwoerd, let's move on a little bit. I think let's go back to Dr. van Zyl. Slightly off the topic, but there's a lot of uncertainty of how the virus transmits. How contagious is it really? And what role a potential carrier could play? And I think we can cut back to some of the other guests or panel members if you feel uncertain about that portion. But how contagious, how is the spread of the virus? We're looking at vertical, we're looking at horizontal spread, and if you could maybe enlighten us a little bit more about that.
Look, we know that the virus spreads through feather dust and so on, and this is just actually a continuation from Prof. Bragg's conversation about the, quarantine facilities. Feather dust, we can't destroy the virus, we can't kill the virus, so it sits there in the little nooks and crannies of your cage, right, or of your aviary. I'm sorry, someone's playing really loud music in the background here. So it's really hard to destroy and really hard to monitor that. And if you have a susceptible bird in some way, shape, or form, it's probably going to catch that virus. I speak under correction, but I do think there is some vertical transmission as well. And, again, very difficult to control when birds are asymptomatic and difficult to predict when birds are asymptomatic. But I do feel that the virus is quite contagious, which is why we're all putting in this effort to find any sort of solution, whether it be containment, whether it be vaccination, and even testing, because testing gives us information with how we can separate these birds, and then we can track birds that we know are 100% positive, and we can see how they behave, and we can do like more targeted work to figure out how resistant is the virus, how transmissible is the virus, and exactly in what mechanism it transmits. And I think Prof. Bragg can also speak more to that.
Okay, it seems like we've restored our link. No, it seems like we lost it. Well, he can't hear us. Prof. Bragg, would you like to add something there? Especially because there's a lot of uncertainty of how to approach the virus. Will one pair of birds in an aviary affect the pair next door? How important is species? Because certain species tend to be more susceptible, or we presume that. And I mean, even came a question that what is the effect of wild birds on the spread of the virus? So let's dwell a little bit on how the virus spreads, and can a bird develop immunity? What age groups are more susceptible? If you maybe just add something like that while we try to connect with Dr. Verwoerd.
Okay. In my experience, I've got a terrible echo. Have you got an echo?
We can hear you have a slight echo, but ours is fine.
Okay. The virus is highly infectious in the chicks. So in the hatchery setting, it spreads very quickly between the chicks. In the adult birds, it appears to be that they're more resistant. So it doesn't spread that easily amongst the adult birds. And then obviously there are species difference. The South American birds appear to be less susceptible. I've got a terrible echo, so I'm going to log out and log in again.
Rob, you seem to be better. Try again.
Can you still hear me?
We can hear you nicely.
I can't see you. I don't know what's happened. I'm not hearing the echo. If you can hear me, we can carry on. The South American birds appear to be less susceptible to the virus. In fact, when we were doing the hemagglutination tests, so that's the ability of the virus to bind to red blood cells, we found that the virus didn't hemagglutinate with, for example, macaw blood. So the South American birds appear to be much more resistant. The Australian, the Indian, and the African birds are highly susceptible. I think the Australian birds are probably the most susceptible, followed by the Indian birds and followed by the African birds. I've complete. As long as you can hear me, I'm sort of seem to be can't see myself here. So while you're carrying on, maybe Alta can comment. I'm going to see if I can log in again and get sorted out.
That's great. Thanks, Prof. Can we cut back to Alta quickly? Is there something that you could add for us there?
No, I mean, for us, it's very obvious that the virus affects our native birds in South Africa. We just have to look at the Cape Parrots, right? And they were on the red species list for endangered species. So again, this we need to push for all the measures, like in a holistic approach, not just vaccines, but everything else. I'd also just like to make a comment on something Prof. Bragg mentioned earlier. In South Africa, it's getting hard to do some of this research even. We've got so many regulations, even at a university scale, with all of the permissions we need to work with live animals, to work with the blood and the serum and GMOs. And so there's a lot of regulatory hurdles, and they start like from the bottom of the food chain. So it's been quite hard, and it's been a very paper pushing game as well in terms of development of even developing a diagnostic. They have to utilize serum. So getting those from the Department of Land Reform, it has a compounding effect on how we can push this forward. And then obviously also the funding landscape is always a problem. We've been really lucky in that we've gotten funding from quite a few institutions and bodies over the years, but it's always hard to future-proof that development process because you don't know when your next tranche of money is going to come in, and also what regulatory hurdle you're going to hit at the end of the day. So it's been quite a juggling process, all of this research, all of this development, but we persevere because we feel very strongly about it. And I think at UCT especially, we had some really exciting vaccination results last year. So it would be great to see this work go forward and some more development going into it.
Thanks very much, Alta. I see Daria would like to make a comment on the virus. Daria, you up? You can.
Yeah, this is a little bit on the testing side of it, but we have tested in, I think we've tested a macaque positive in December. So I agree that the susceptibility might be less. However, it's still present, and it still affects the birds. And then just another comment on something that Prof. Bragg said on if the virus isn't in the blood, then it can't be detected. And I agree with that. However, with tests, I see that they haven't really distinguished between PCR and real-time PCR tests. So with if you use a test that's more sensitive, even if the virus was in the blood and it circulated and the body got rid of it on a natural way, it breaks down the cells, that DNA is still present in the blood, and we can still detect it on our side.
Thanks very much, Daria. We're cutting back to Prof. Bragg. You seem to have recovered the connection. Just I'd like to add a further question that's popped up is, with, for instance, other viruses, we've developed treatments, antiretrovirals in the case of HIV. Would it be possible to develop a similar sort of product for this virus, or because of the virus's structure, it would not be possible?
Yeah, I can comment on that. But I first want to make a quick comment on something that Alta said about you can't get rid of the virus. The virus is extremely resistant. There are very, very, very few disinfectants that will actually work on it. But having said that, and you might hear in the background, you might hear 2 parrots screaming. I've got 2 pet parrots at home. And during all this time, I was working with infected birds, isolating, or not isolating, but purifying virus, working with the birds, handling sick birds, constantly, day in, day out, day in, day out. I made use of good biosecurity. I made use of a good product that inactivates the virus, and my parrots at home are still very happy, didn't pick up the virus while I was working with it. So it is possible to remove the virus. But to get to your question on antiretrovirals and other options, viruses, we all know about antibiotics. So antibiotics are, you have a bacterial disease, and in the good old days, an antibiotic would work. Now we have major resistance problems. So you could have an antibiotic, you don't have to know what the virus is, what the bacteria is for the antibiotic to work. The problem with antivirals is they extremely specific to each virus. So you have to understand exactly how the virus is working, and then you have to find a way of blocking some stage in the life cycle of the virus. So again, because we can't culture the viruses, makes life very difficult, and it's very expensive to make an antiviral medication. The other possibility could be looking at antibodies. You could perhaps make a panel of monoclonal antibodies that you could use as a treatment option. So if the birds are get infected, you inject them with antibodies that will help to rid the bird of the virus. You could also try and look at producing antibody fragments. We just finished a project where we looked at using a phage display library. We were able to find a short peptide that has anti-Newcastle ability. So it actually binds to Newcastle and inhibits it. So it's possible. And we were working with Ed and the plant people to try and see if we could get this anti-Newcastle peptide expressed that we could see if we could actually go further with it. So something like that would be a possibility to look at alternatives that might be viable. But that needs further discussion, and we can, get young, motivated researchers like Alta and get some funding, and I can be involved in assisting in the background. We could look at doing stuff like that.
Thanks very much, Prof. I think we can summarize up until this point is that we sit with a virus that is very difficult to work with, mainly because it can't be cultured. 2, because it is very, very resistant to many of our known disinfectants. But a lot of questions arise concerning how do I manage a situation in my collection where I have a suspected positively beak and feather disease bird, and what will the influence be on the other birds in my collection? Do I have to stop breeding? Can I manage it? And I think we'll probably on section 3 we'll be able to get a bit more into that. But being scientists, we tend to see the extremes in everything. But how, on a practical base, how serious is the virus in our collections? And we see we don't have a connection with Dr. Verwoerd back yet, do we? Okay. I think what we're going to do now is maybe just leave this section pending, and then while we come up with the difficulty of testing for the virus and tests involved, we'll bring up our different, we'll go over to the next section, which is on the laboratories and the testing procedures and techniques. So we'll have a quick cutaway and allow some of our sponsors to talk to you, and then we will start with that next section. Thank you very much, and please keep on letting us know where you are from. It's always nice to see some people that are involved. We have a guest from Kazakhstan. That's great. Right, welcome back, everyone. We're now going to give our participants from the laboratories chance to explain the different testing techniques, the tests that are involved, how we go about sampling correctly to make sure that we get a reliable result. And I think I'm going to start by asking Dr. York to give us a bit of background on the different tests that are available and the techniques that are used in the laboratories. After Dr. York, can you please unmute? I won't hear me to unmute it. Well, yeah, okay, great. Dr. York, can you please unmute your computer?
Sorry,
Thank you.
I didn't realize that I had muted that. No, thanks very much, and for interesting first half of the session. Our focus, we're obviously a molecular diagnostic laboratory, so our focus is on the molecular tests. When I use the term molecular, I'm referring to PCR, and the PCR in itself, they're very various forms of it that have evolved over time. You've got the qualitative PCR, the quantitative PCR, the real-time PCR, the nested PCR, the hemi-nested PCR, the isothermal molecular tests. But the value of the PCR technology and the other molecular-based tests is that the assays can be designed to be extremely specific for the genome. So the challenge is obviously to find that region of the genome is conserved amongst all the species. In that way, you are sure of picking up the virus. The challenge, however, is that, and whilst we have the benefit of the research that's been done in the past, sequencing the many genomes over the years, that the primers have been designed to target those conserved regions. However, it's a very small virus, the smallest around there, around about sort of 20 nanometers, small genome, but it mutates very frequently and recombines. So there's always that challenge in the back of your mind that a negative PCR in the one lab could be due to possibly a difference in the primer sets that are being used. But whilst and you can look at the sensitivity of your test, and you can do endpoint titrations and say your PCR has gone down to single copy per reaction. But the one of the drawbacks and limitations that we've had, and we create our own sort of standards, is that there's no international not that I'm aware of, international reference, characterized international reference standards, so we can measure that against. So we can say, oh, our PCRs are excellent. However, when you look at the literature and you read about it, the virus itself is influenced by the viral load, the PCR, the viral load, the stage of infection, the site of replication, and the shedding of the virus. So whilst we, in the beginning, we really focused on looking at encouraging people to send blood in, because there's two parts to the test. The one part's the lab where you can control everything. The other part is the sample that is sent to you. Now, if that's not carefully taken, and I was just going to go through a little slide to show just highlight a few little points that one needs to be careful of in taking the samples, because the way that sample is taken in the field can also influence the result that one gets in the lab, the cross-contamination and handling infected birds unknowingly, not being very careful taking the samples. So shall I carry on? I'm just not sure when I should actually stop talking.
Thanks, Dr. York. If you can submit that slide about the sampling technique in the field, we can post it on the link. Yeah, but we'll come back to you now. I think
you help me do that, actually.
Okay, we're going to get the technical guys to help you with that. In the meantime, I'm going to cut across to Dario to explain to us the difference between PCR testing and real-time PCR testing.
Yeah, these are basically the two that are used the most in testing for beak and feather, and it's simply because it's been used for quite a long time. The primers have been standardized to a certain point. Yes, there is mutation, but another point that when they're designing these primers, it's normally for a functional, not just a conserved region, but also for a region that's functional to the virus. So if that mutates, then the virus won't be functional afterwards. So for a virus particularly, the viruses don't normally mutate in those regions. So the primers are designed for those very, very specific regions. If you take PCR versus real-time PCR is just you run your primers through different cycles, and through every cycle, the section of the primer that you're looking for is duplicated with every cycle. Now, in normal PCR, you look at that on a gel, it forms a little bit of a band if it's positive. And then with real-time PCR, you actually look at a screen as it's duplicating, you see an amplification curve. And real-time PCR, if it's designed properly, is normally more sensitive than normal PCR. So I'm not sure what other methods the other labs are using. At Lumigen Laboratories, we opted for the real-time PCR. It is a little bit more expensive, but more sensitive and more reliable on our side.
Thank you very much, Daria. I'm going to now cut across to Christine. And maybe if Christine can elaborate a bit on the sensitivity and the specificity of tests, what's the difference and how do we measure it? Daria was referring to that, there's the parts of the virus that we are checking for can vary, and that the possibility of mutations will have on test results. Will you maybe elaborate a bit for us, please?
Yeah, sure. So the sensitivity of a test is related to how much of the virus you can pick up, whereas your specificity is related to the specific virus that you're picking up. So it won't necessarily pick up a false positive because your parrot has avian influenza. Your PCR is supposed to be so specific that it only picks up the beak and feather virus. And like Daria said as well, there is the conventional and the real-time PCRs. In SMT, we also use the real-time PCR. We use a commercial kit available. That's just easier for us. And yeah, so the PCR takes a small part of the DNA and makes millions of copies of it. So scientists can study it more easily. And it's like trying to find a single word in a giant book. And the more copies you make, the easier it is to spot. So the real-time monitoring, unlike traditional PCR, which only tells you at the end if it is positive or negative, real-time PCR allows you to see the results, like Daria said, on a screen as it is happening. So you watch it grow in real time. The difference is that it has fluorescent markers and it to track the copies that are made. And when they attach to the DNA, they create like a glow. And the more DNA there is, the more DNA is present in the sample. So that would also create kind of an idea of how much virus is present within your sample. I don't know if I've answered all of your questions.
Yes, thank you, Christine. Maybe we always hear about a false positive, possible causes of that, and can we get a false negative?
Yeah.
Because that is just as pertinent.
Yeah, so with, I'm going to quickly just then touch on the limit of detection. So like with all tests, there's always a percentage that may give either false positive or false negatives. The limit of detection in real-time PCR is like the smallest amount of target DNA or RNA that the test can reliably detect. So false positives happen if there is background noise. So if your sample, like Dr. Dennis York said, if your sample is not taken correctly, and there's a lot of background and you've possibly touched feather dust or anything like that while you were taking the sample, because real-time PCR or any PCR is very sensitive, you might actually pick it up. And that can create a false positive result. So it's got very... there's various stuff that influence it, but then also there's primer dimers that create, like if you're running a PCR too long, you can create false reactions and it binds to non-specific parts of whatever is in your reaction. But there's also a limit that we run the cycles on to prevent any of that from happening. So false negatives obviously happen if there's inhibitors. So like if you have something strange like oil or anything that might affect your PCR, you won't properly extract the DNA and you will get a false negative. So if you think of it as like a fainter signal, the test can pick up and still be confident what is detected in real is real and not just background noise. There is always a possibility that the limit of detection of a PCR, the background noise can create false positives, and you might not be able to detect a virus even though it is present. So, yeah, there's definitely, and a lot of it has to do with sometimes it happens in the lab. It's not impossible, but sometimes it's also the way that the sample is taken or handed in or given to the lab.
Okay, we as soon as we have Prof. York's or Dr. York's slide, is it available? Okay, we're going to put it up quickly, and then we ask him to comment on the sampling technique. Then we'll get back concerning some of our questions around the testing. It's good. Obviously, we can see that no test is a definite until you get a positive, because then unless it's a false positive, of course, if you have a contaminant or so. So we're going to quickly ask Dr. York to proceed with the testing technique in the field.
Oh, can you not get the next slide? Another one? Sorry.
Right, please speak into your mic, Dr. York.
Sorry, the next one. I was just going to.
Yeah, cut across the next one.
Yeah. I was. There were really 3 slides I thought might be useful and relevant. Yeah, this was a slide put together by Bob Philpott in Australia, and when we were doing testing there, and it just to emphasize, not to say that the blood sample is the best sample. It's a good sample, and we prefer it obviously in the lab. But what I realized early on when we started many years ago offering the service is that we needed to try to control the sample that's taken. And a few points here that can also result in sample mix-up and incorrect results. The labeling of the tube. So, and there might be other methods that other labs and people use to collect the sample, but labeling of the tube is extremely important. Collecting the sample, this was sort of done for sexing. He's an author, but he also is a breeder. And just to go through the process of taking the sample, we found that taking a little blood spot just above the claw, following the needle into that skin just above the claw, and then pausing, and then taking the sample. But on each of our kits, sterile kits, we have a little marker there. You'll see it there. He took the sample very nicely to illustrate the amount of sample that's taking, because that also can influence it. You know, some breeders put a very small amount, and obviously, even if you send the same sample to 2 labs, one's got a little spot and it's a low level of detection, a low viremia. Another one's got a whole paper filled with blood, which often comes. People think they need to take more of the sample. If you can just keep your blood spot just below that line, that and try and get to that as best as possible. But in principle, if you can see a clear spot of blood, that's sufficient. And then place that back into the tube, but always holding the little spit, the little bit that sticks outside the tube. And then documenting as clearly as you can the label and this bird's identity. We have a very nice system that you can then go on and log your information onto. It's an online website that keeps your results, or always your history there, and it's very useful from a testing point of view. But if you could go to the next slide, because I wanted to illustrate there are a couple of faults in that slide that you probably all are going through your mind. Ideally, when you're collecting a sample, 3 people should be involved, all gloved and masked. The more protective clothing you put on, the more conscious you are of the importance of it. I think a good analogy is think of beak and feather like a COVID virus. You can't see it. There's millions of copies per microliter. It's just so much virus around, secreted through the secretions, through the feces, through the infected feathers. So if you have it there, you want to try and reduce the spread of the virus from bird to bird, but also to take a sample carefully. I don't have experience in the other disinfectants, and I'm sure there's a lot of work being done. It's just in our lab, bleach is our friend. Bleach, we bleach everything, but the key with bleach is you've got to leave it on the surfaces for 10 minutes and allow it to take its action. It destroys DNA. I know, as Prof. Bragg said, it's an envelope virus. It apparently, I haven't done the research in that, it neutralized the infectivity of the virus. But others, Viracle, Burcon, I'm sure they all work well. It's just, this is the one I'm familiar with. Whatever you use, bleach your surfaces. And then the second person is the catcher and the holder. Again, he goes, gets the bird, returns the bird to the site, new gloves at each bird, and he holds that bird out, the claw out for the bird pricker. The bird pricker then pricks the claw. Again, he uses new gloves for each bird. And then the sample collector is the person who does the labeling, never touches the bird, never touches the sample, and he removes that little bit of paper and touches it against that blood spot. There's no contact with the bird, puts it in, no contact with the lower part of the filter, and then puts that in, and then sends that off. In this way, you reduce the spread of the possible cross-contamination from an affected bird to the next bird. And don't take too much sample unless there's a specific reason for that. And yeah, that's what I wanted to say. And the next slide, I used this again to illustrate, you know, we have many real-time accredited real-timers, PCRs, and we know the technology well. But I've used this to illustrate the point that Christine mentioned and Doris might have mentioned, is the limit of detection of a test. So here's just a titration, taking 5,000 copies of template, just taking purified as pure as you can, be it a plasmid or what, and you titrate that, work out the DNA concentration, the number of molecules you have per microliter, and do a dilution series here. So this top band, what you're looking at, is a PCR product. So your positive control on the right-hand side, just look for that band. So this is just the amplified DNA that Christine was talking about. And what you should see is as you do this titration, your 10-fold initially, then a 5-fold titration, these are copies per reaction, and you're going down, and you should reach an endpoint at some stage. And this area here, because the question that was also asked is, what about the weak positives? What does that mean? Well, when you're testing at the limit of detection of the test, there are a lot of factors on there, the sample volume, how, you know, you're working in very minute concentrations. So this area, this weak positive, it's just a warning to say, just, you know, it's very low. I use the analogy of like if you had to take a bucket of apples and put in 10 oranges, and you say every time you put your hand in, take out an orange. Well, you would, when there are 10 oranges there's a good chance that you'll pick out you'll be able to feel for that orange. But as you go down to 1, every time you put your hand in there, you're not going to put an orange in. So we've taken this endpoint dilution, this lowest bottom here, and if we go to the next slide, here is that same sample, just repeated exactly the same sample 4 times. So you'll see here, just to illustrate the point, at the very low viral loads, you can get one, like sample 2, you're seeing nothing there. So if that sample had to be sent in, you might get a negative sample. If the one on the left had to have been sent in, and you sent the same sample in, or a duplicate, those 2 samples, you might get one positive and one negative, and you'd say, well, look, what's the problem? And as Prof. Bragg mentioned, is when you've got this intermittent shedding and these low viremias, you know, in the initial stage of an infection, my understanding is if a bird gets infected in the feather and the active replication is the hair follicle, your focus is going to be, the viremia is going to be there. So taking the blood sample at that stage, you might not pick it up. Why? Because there's no virus there. But the feather is where the virus is. So, and I mean, there are lots of studies, more and more recent studies, I see they're looking at the feather. So you've got to be careful of, sometimes another sample might be an important sample. For example, a cloacal sample might be, shedding of the virus might be more significant there. But an oral sample, if there's oral shedding, might be important. So you've got to bear this in mind. And sometimes, and this is where one works together, you need to work together with the client, sometimes sending combination samples. But again, taking blood is a little bit easier in my view. Taking feathers, you've got to be very, very careful because the risk of contamination. In the lab, I must say, we don't like feathers, especially if you start to send 3 or 5 feathers in a plastic bag. Everything gets dealt with in a class 2 hood. And you know, you've got your fan there, those feathers go all over the place. So you've got to weigh those up. And ideally, if that is your case, we'd like to go through, try and use that tube, cut off the blood feathers, put your blood feather into that little tube and send that in. But again, wear gloves, be careful. Or if you're taking a swab, cut the end of the swab, put it into that tube and send it to the lab. But just make note, look, we've got a feather pulp in there and we've got a blood sample, or we've got a swab sample.
Thank you. Thank you very much, Dr. York. I think it was very insightful. I think you've answered a lot of questions that people have had. I'm just going to refer back to Daria. If I understood, I'm not sure whose comment it was, that different laboratories might be using different reagents from different DNA particles or different virus strains. And that could also have an influence on test results. Did I understand correctly?
Yeah, the different strains can definitely have an effect, but in short, the primers are designed to get a universal region, or a region that is covered in most of these strains. So I don't think, especially I think SMT, they said they're using a commercial kit, then that means those companies should make sure that it covers most of these strains, and it isn't a universal region that has been sequenced in all of the different strains.
Thank you very much. Can we cut through to Christine? She would like to make a comment, and I would like to add maybe a further question after a comment, is that must we start looking at other testing techniques like Dr. Bragg referred to?
Yeah, so I just wanted to make also a comment on the primers. So when we started testing beak and feather, we had various different primers that we tested, and we got variable results as well. The reason we went with a commercial kit was because it had the most consistent results. With the other designs from different articles gave us very strange results, and we were very unhappy with it. So we chose the ones that were best for us and best for the industry at the time. Obviously, like Dr. Anton Smit said, that there can be mutations, and with that as well, you would also get variable results if different labs are using different primers. So what the DAH does with avian influenza is they make sure that every lab uses the exact same primers and probes to make sure that there's consistent results. Obviously, that's a notifiable disease, so they have control over that. I just think that a collab with the industry to make sure everybody's testing the same type of thing would probably be also the best thing. With proficiency testing, we also do and make sure that everybody tests the same type of sensitivity, and that your PCR is like on par with the rest of the world. The only difference is there's no proficiency test available for beak and feather, which is really a pity, because it's a very big problem in the parrot industry. So yeah, that's what I want to say.
Thanks very much, Christine. The use of, for instance, ELISA tests, should we be combining it with our PCRs? How do you see the future to try and get more meaningful results?
I think that depends on what your end goal is. So if you're testing your bird to see if it had a past infection, serology is a great option. There are ELISAs available to detect the pathogen. I don't really know how sensitive they are. I know for Salmonella, there's definitely one that is very sensitive. I'm not too sure how far people have delved into that type of thing. But if a PCR can pick up the infection in the blood, I'm not sure if the ELISA will pick up the infection in the blood if it's not there. So that is kind of my opinion about it. I think maybe a combination of it. So if you're looking for a live infection, you know your bird is sick, PCR is definitely the best option. But people trying to convince me that an ELISA test is maybe better, I have different opinions about it, because a PCR is for live infection if you are currently having a sick bird, or it is to eliminate the fact that you do have a sick bird. And with this virus, it's very up and down, because you might have a carrier or you might have one that's currently infected, but it's asymptomatic. And then you're blaming the lab for a false positive, but your bird does actually have beak and feather. So the virus is very irritating.
Thanks, Christine. I think you summed it up very correctly there. Prof. Rob Bragg wanted to comment, and then Daria after that. So we'll cut through to Prof. Bragg first.
Yes, I think clearly there's a difference between testing for antigen and testing for antibody. When you're testing for antigen, you're looking for the virus. So you're looking for an active infection, which has a very important role to play. When you're looking at serology, you're looking at history. So you're looking at, has the bird been exposed to the virus in the past? So the drawback with serology is it takes at least 2 weeks after exposure before the birds have antibodies. So you can't use serology in a disease outbreak. But when you're thinking about auctions, if there's a reliable serological test, you can test if that bird has been exposed at some stage to the virus. It could be an asymptomatic carrier. So the PCR test could test negative, but yet it had been exposed. So then the buyer can decide if he or she wants to take the risk of buying a bird that has got a history of exposure to beak and feather. So I think there's definitely a place for a serological test. But again, this virus is fun to work with. It's not going to be easy to do a serological test. You have to do a competitive ELISA. You can't do an ELISA using conjugated anti-chicken. It does not work. So you get a signal, but it's pointless. It's meaningless. The only ELISA that would work would be a competitive ELISA, where you set up the ELISA so that the virus, if there's antibodies in the bird, they bind first, and then you put your reporter antibody on, which if there's lots of antibodies in the parrot, they can't bind, and then your detection is on the reporter antibody. And that works well. Competitive ELISA works well. But there's a number of things that you have to have in place. You have to have antigen to coat the plates with, which is doable. You have to have a good supply of beak and feather specific antibodies that are raised in something like a chicken or a rabbit or a mouse or a horse or whatever that you can use, and then you have whatever you've raised your beak and feather antibodies in. Then the competitive ELISA works very, very well. But you can't do that on a drop of blood. You need serum. So you need a larger volume of blood, but it's highly doable.
Thank you very much, Prof. Daria, you wanted to add some comments?
No, Prof. Bragg addressed it at the end. We have to consider the logistical issues if we want to go serological. It's very difficult to get enough plasma. It's a proper drawing of blood from the birds. It's not as easy as just getting a blood spot.
Thank you very much. I'm going to address a question that popped up, and we've had it in the past as well, is what is the chances of a rapid test that a person could do at home? Is there any development in this field? Would it be a practical option? And I'm going to refer to the whole panel to respond to it. I think let's start with Daria, and then we work our way through.
So in terms of rapid testing kits, there's been a lot of research being done, not specifically to beak and feather, but just the technologies that have gone into it to make it cheaper and more affordable, because most of these rapid kits cost more than a real-time PCR kit at our lab, for instance. So we have to consider that, but I'm not sure about the development of one specifically for beak and feather. It sounded like Prof. Bragg had some experience with that.
Can we ask Prof. Bragg for his input?
No, I don't have any experience with rapid testing. A lot of the rapid type testing would be serological testing. So if you've got a serological system that's working, then you could maybe look at a sort of cage side test. But at this stage, it's a very, very long way off.
Dr. York?
Well, we are involved in the rapid tests, and I've been amazed. The first tests that came out were antigen based in the poultry industry. Not the first that came out. I mean, HIV was the leader in terms of the tests. There are exquisitely sensitive antigen tests at the moment. So they start off as being colloidal gold, then they move over to fluorescence, and then they become more sensitive and more quantitative. There's a linear curve in terms of the amplification between concentration and the intensity of the So there are antigen assays available already. We have them, and it won't be long before they do come out with, if there's enough demand for it, a rapid point of care kit. What Darius also was mentioned, he was referring to the rapid molecular test. I mean, there are rapid molecular tests, and it's possible to put those up. There's obviously going to be limitations, but I think if you had to compare the application of a rapid point of care test, not a molecular test, I think the PCR will still be the one that will need to be done really to give you that ultimate sensitivity.
Okay, thank you very much, Dr. York. I think we've got a lot of background now on the challenges that the laboratories have and what's involved in the testing. I think as we've all realized, this is I think Prof. Bragg summed it up properly. It's a funny virus. It really is. It's causing us a lot of headaches and fun. But that's why I suppose why we are involved with it. But I think we all understand the challenges that the laboratories have much better now, and the interpretation of the results. and that it's an ongoing process, and that some more work will be done, and we'll progress the whole time. I think we're going to wrap this section up for the moment. We'll have a quick ad break, and then we'll go cut back to, we've got our connection back with Dr. Verwoerd, and then we'll cut back to his comments that we've asked for earlier. Thank you. Welcome back to everyone. I think we're progressing quite nicely. Please remember that this, we have our guest from Iran, Cape Town, Windhoek. It's wonderful to see you all, Postmasburg. We'd like to remember that this feed is available all the time, so please share it. We will also be making clips out of it and making it available through our association. I hope everybody's found it as interesting as I have up until now. It's been very, very insightful. You think you know enough, but you never know enough. And we're going to ask Dr. Verwoerd to please comment on the questions we put forward to him, which you unfortunately weren't able to connect with him. So when we go cut back to him, I see he has been restored. Back to you, Dirk.
Right, hello, Anton. Yes, I moved sites and I'm connected now by the phone. There's some issues with where I was and the computers and so on. So I missed out on the last 10 minutes or so. Can you maybe repeat questions that I must comment on, or how do you want me to progress?
The question was you had quite a bit of field experience about the spread of the virus and how that affects the immunity, do birds develop immunity, and what was your experience with possible mutation of the virus?
Right. I think the first thing that has to be said is that the outcome of any infectious disease situation, viral infections in particular, does not only depend on the virus or the presence of the virus. It is the classic epidemiological triangle. So you've got the agent, you have the host, and you have the environment. And even if the agent is the same agent, same Psittacine circovirus that we're talking about, the host is different when it's a very young chick, when it's growing, when it's going through the predictable stress bottlenecks of feathering, of maybe weaning, being exported in a container with in an aeroplane, with birds from every description, all of those things. That's completely a different outcome than, let's say, an adult breeder in a secure, stable breeding establishment, or even a pet bird. those environmental variations as well as the host elements, and you can carry on with that. Your nutritional deficiencies. I've investigated these things dozens and dozens and dozens of times. Mass mortalities in young chicks very often turn out to be something completely different. But we have this preoccupation that any high mortality must be beak and feather, or any feather abnormality must be beak and feather, or any feather loss and so on must be beak and feather. That's just not the case. So I think the first element to understand is this interaction, which means that we need a holistic approach, which means that we have to do proper diagnostics. And that is normally my first response when I get any inquiry. Let's get to a proper response. Send me material, get in touch with your veterinarian if you're far away, do a proper investigation. We have to look at more than just immediately jumping to the conclusion that it's beak and feather.
I think, Dirk, it's important here to stress the fact that one of the main, I won't say symptoms, but results of beak and feathers is immunosuppression. And quite often,
Correct.
Loss of birds is not directly due to the beak and feather, but to secondary complications. Could you maybe elaborate a bit?
Yes, absolutely. That's very, very true. And there's also, I'm sure someone else has mentioned about carrier states and latency and so on, recrudescence. So it can be an early infection, could be vertical infection, could be mother to child type of scenarios, which does not present clinically at all, and then comes through later through these predictable bottlenecks, and then it just explodes, and it explodes in different eventual modalities. So we always talk about an ultimate cause of death, but we're really interested in the proximate cause of death. What was the leading cause? And this is where the important perspective of beak and feather lies. Not only focusing on the clinical thing, but do a proper diagnosis, and then you'll sort of unravel the process. Definitely immunosuppression being the most important presentation. That's why I think the name, we all realize that is a misnomer. It's a historic misnomer. And the beak and feather and so on abnormalities represent a very small part of the picture. Immunosuppression also means that you have to look at stress management. You have to look at nutritional aspects. That for us that's very, very important. We've seen that time and time again. Critical nutrient lacking, and then
Okay, we're just going to try and reconnect to Dr. Verwoerd. We just had a question also, it's popped up on the screen that I think is pertinent to what Dr. Verwoerd was saying. Is the question was, is beak and feather seasonal? I think we could maybe refer to Prof. Bragg to maybe make a comment there while we wait for Dr. Verwoerd to come back.
I'm afraid I don't have enough experience with beak and feather in the field to comment on if it's seasonal or not. Most of my experience was working with the infected birds that we got in the lab. So I can't give a comment on that.
Did you find higher levels of viremia in certain conditions, for instance, molting season? Would that be a possibility?
No, not really. Look, we kept the birds. You know, the birds that were kept at the University of the Free State were sort of, gave them the best housing we possibly could, but there was obviously a fair amount of stress involved. So the birds, we were handling them quite often as well. So there was lots of stress that could have induced viremia. But it's I can't really comment on the seasonality of it. I'm sorry.
Thanks very much, Prof. We see we have Dirk back. Dr. Verwoerd, a question popped up in the meantime, that is there a possible seasonal tendency of beak and feather disease?
I think the patterns that we observe is more related to the seasonal tendencies in breeding, the breeding seasons, the bimodal breeding that we often see with African greys, for example. So those things represent a seasonal presentation of the infection. It's not really about the seasonal, let's say, more common, less common, whatever of the virus. It's about the host dynamics, which gives us a seasonal pattern. Okay,
a further question that was asked earlier was how how contagious is the virus in a practical sense? I mean, remember, we scientists see statistics and data, but in the field, in an adult population, at what rate would it spread, or could it spread? We heard that the virus is very, very infective and it's very resilient. But in a practical sense, what is the spread rate within a closed population?
Yeah, so again, it's about the basics. The basics is level of challenge and level of resistance. And resistance is not only the specific or antibody mediated and cell mediated and so on resistance, it's also the general, the non-specific ability of the host to withstand infection. And that goes back to, the nutritional elements and the stress elements and so on. So it's always that balance. And therefore, the answer to that question will differ from situation to situation. It is true that I think it's an extremely infective agent, very contagious. And of course, as I'm sure many people have stressed, it's really difficult to kill with a disinfectant. And you have to make sure that you do your disinfection, from every angle as good as possible. So it really will be different. We've investigated outbreaks where it seemed to run like a wildfire through a breeding population. And in others where only certain individuals were infected. I think it's part of the solution to try and drill into an element where you find the disease factories. I do this in all hosts, in all animals, in all production settings. There are always individuals that are so-called super shedders or intermittent shedding and constantly feed the fire, so to speak. So it's important through testing and breeders, we normally test the chicks to find those birds, if it's possible, cull them. If not, if there's genetics and other considerations involved, then try and move towards a dirty and clean flock situation, but try and make a separation between what is clearly infected and what is then clinically not infected. So we will find a situation, and we see this in many hosts and many infectious agents, that there is a balance that develops in a large percentage of the population, but certain individuals definitely are more susceptible or, as I said, are higher shedders.
Thanks, Doc. One further question is, would it be possible for a bird to heal or recover from an infection, or will it always remain a possible carrier? Does it, and can they develop immunity to this disease?
Yeah, they can definitely develop immunity. There's no question. And remember, immunity does not have to be the complete absence of an agent. It just means that it's kept at bay. We have many, many examples in veterinary medicine of animals that live comfortably with an agent, whether it's a parasite or blood parasite or a virus, whatever, latency, herpes virus has come to mind in many species, even in humans. And, so it's not about absence or presence. It's about what the balance is. So yes, there is an effective immune response that is possible, both with vaccines as well as natural exposure. And it's a matter of keeping that at a level that the virus doesn't win the war, and you get clinical manifestations, or that the virus is then passed from the hen to the chicks, and the chicks then become clinically affected. They die and so on, while the parents remain 100% clinically healthy. So all of those scenarios do occur, and this is where we have our control points, and this is the exact detail of how you position your vaccine intervention and your biosecurity and so on.
Thank you very much, Dirk. I just have a further question, which I'd quickly like to put through to Dr. Alter. If we can get her up again, please. Alter, immediately the question arises, are there any other viruses that could cause increase in beak and feather disease virus? Because we quite often know that these viruses run hand in hand.
Yeah, that's true, but I am going to lie if I give you an answer here. So I'm not 100% sure, to be honest, that is not my area of expertise. But like, Dr. Verwoerd said, there are other circumstances we need to consider when these birds become ill. And if they're immunocompromised by some other disease, whether it be viral, bacterial, fungal, it might have an effect on the disease progression of BFDV.
Okay, thank you very much. While you're on the line, there has been a vaccine developed, either I think it's been experimental in Australia. Do you have any knowledge of it? Could you maybe add something for us there?
No, I must admit I don't have the finer details of that vaccine, to be quite honest. But I would suspect that it was probably targeted to the coat protein, if I'm not mistaken. But I do speak under correction.
Can I maybe ask Dr. Verwoerd if he has any knowledge of it?
Yeah, not firsthand, just what's in the literature. And yes, of course, they did fantastic work. That's Professor Rydal and his team. And they did focus on the coat protein, the capsid protein, just like we did. And I think everybody, that it being such a simple virus, you've basically got two proteins to target. It's either the coat protein or the replication protein. So everybody goes for the coat protein. That's the outside element, you can say, that presents to the cells and of course, being presented to form antibodies against it. So yes, I'm not sure how far. There was a provisional notice that it's submitted for registration in Australia. I'm not sure if that went through. The motivation, of course, for them is almost 100% about the conservation value. They've got highly endangered parrots that are really susceptible and under threat. So that was the big motivation. And unfortunately, in the day and age that we live in, conservation only does not generate the funds to do this kind of development. We have to balance it with a commercial agenda, and this is what we try to do. We try to balance those elements, and I think if we look into the future, we might see a range of different, even capsid-based vaccines, because the Australian parrots are, of course, different to the African parrots, etc. So we follow what's being published, and I just acknowledge the pioneering work that group has done. But that's what I know.
Thanks very much, Dr. Verwoerd. I think we're going to wrap up the first two sections because they are very intertwined. Is there any of our panel members that would like to add something, make a comment before we move on to our next section? Christine shakes her head. Prof. Bragg, nothing. Everybody happy that we've dealt with this section completely? Thanks very much. I think we'll now move over to the next section, but before we get there, the next section will be the way forward, and then we'll have the whole panel involved. We're going to cut through to our sponsors and advertisers. Thank you. welcome back. I see we're keeping a tight time schedule. We are now engaging on the way forward, and I'm going to give each of the panel participants an opportunity to briefly give us an indication of how they see the way forward with this disease. And I think we'll start maybe with Prof. Bragg, if you don't mind.
Okay, thank you. I think there's potential for various molecular-based vaccines. I think the work that has been done at UCT in the biofarming group, where they're doing some amazing work, could lead to potential vaccines. The problem, as I mentioned previously, the problem is vaccines need to be registered, and they're also GMO. So we need to look at the way forward with that quite carefully. There's probably would be beneficial to investigate a serological test. This would then potentially limit the spread of viruses through the auctions. So I know the auctions are a critical part of the industry, a very important part of the industry, but it's also a very good way of spreading the virus. If you can have a PCR test as well as a serological test, I think that would be beneficial. There's also the possibility to look for producing antibody fragments or maybe antiviral peptides. Like I say, we were successful in doing this with Newcastle. We just got to a point where we were working with Biofarm and we didn't get the expression that we were hoping for. But the project stopped basically because the student was finished. So looking at something like that, where you can look at maybe developing an antiviral peptide or some sort of treatment option is a possibility. There's a variety of technologies that can be looked at, provided there's funding and bright young stars to take it forward.
Thank you very much, Prof. I think we'll give Dr. York opportunity to give his opinion. Dr. York, your microphone is muted. There's no need for you to mute the microphones. We'll mute it from our side.
Yeah, strangely, I don't recall doing that, but yeah, look, I think the goal of PCR is a wonderful test, and whilst excellent research is going and progress has been made, vaccines should be the goal forward. More rapid tests will be useful for the veterinarians and for the breeders, but the molecular test at the moment is the best tool to manage beak and feather. And the goal of every owner and breeder, it's been mentioned earlier on by many, is to obviously have a negative flock. That's your goal. Testing plays an important role to establish your baseline. It's been mentioned about taking your birds out. If you take your birds out of your environment and you've established a negative environment, treat them as if they're potentially infectious coming through. They must be isolated, quarantined, tested. Preferably, try not to bring them back into your breed. Focus on your breeding flock, testing, establish that using testing and regular testing. Where you have a problem that you feel the test isn't correlating with what you expect, especially clinical signs, bring that to our attention, because it might be worth looking at another sample type. Take a feather rather than just sending the blood. Look at a fecal swab or look at an oral swab. Test those other samples to assist you. But regular testing, delayed, continuous testing is going to help you manage it. What you want is to get, the goal must remain a free flock. Using of the detergents, as I see in the lab for contaminants, that dilution effect, wash, dilute, just dilute the antigen, wash the virus, wash.
Thanks very much, Doc. I'm going to ask Dr. Verwoerd to give his insight into the future and to new research.
Oh, thanks, Anton. I fully agree with both previous comments. Absolutely necessary. What I do see a lot is people try and build an entire picture on a single test. I'm sure that's been mentioned. It just doesn't work. A low titer or a low cycle number, is it low going up or low going down, and so on. So a second, a follow-up, repeated testing, as Dennis have said, all of those elements come into it, and it's really important to understand that part of it. And going forward, I think we're in exciting times. We've got proof of concept on a number of prototypes and of a number of different approaches. And for me, I'd like to see the toolbox a bit fuller than what we've got at the moment. And in fact, I see a place for several different vaccines. I see a place for a simple capsid vaccine, expression vector type vaccines to use in the face of, because that's the practical reality we'll have in the face of an outbreak situations for the foreseeable future. And this is where a live viral expression vector model is particularly suited. I also see a massive potential for the oral route, and I think this is where the biofarming unit is streets ahead of anybody else. And I'm looking forward. We've got all sorts of plans to go forward on that, because that will address many of the issues where you do not want to handle birds. You cannot handle your breeders all the time. You want to be it must be a hands-off type situation. Even with chicks, rearing chicks, with the rearing foods that we use and so on. So the oral route is really attractive. And I foresee a toolbox in the future with all these available to us.
Thanks very much, Dirk. Can we ask Dario for his inputs, please?
Yeah, it's also been exciting for us to see what the breeders have been doing, because at the highest, I think we had a 24% positive rate of testing at the lab, and over the years it's gone down. I think at the moment it's 12 or 13%, which is a lot better. However, a big problem that we've seen is that we've mainly been receiving, or there's been an increase in samples from exporters, but our breeders themselves have gone down in testing specifically. So work with the labs to make sure you keep your flocks clean, and make sure that you guys test on a regular basis, even if you are only doing spot checks, or even if you're doing pooling, those kinds of things. We just want to make sure that it's negative on your side. And then make sure, if, like Dennis York also said, make sure that you collaborate with the lab. We're open, we're happy to work with you guys to make sure that we're keeping your flocks clean.
Daria, thanks very much. I'm going to refer back to Dr. Alta van Zyl for her inputs on how she sees the way forward. And obviously, research costs money, and what sort of funding and where is it coming from? Can we expect some help from our our government, our departments? Give us a bit of insight there, please.
Okay, so in the past, we've gotten funding. I'm going to start there because that's the easy one to answer. We've gotten funding specifically for students on the project from the CSIR and the Poliomyelitis Research Foundation. Go figure, right? And then also Parrot Breeding Solutions have been very nice in helping us, the National Research Foundation, and also the Wild Bird Trust. So we've been really lucky to build up little bits of funding all over the place. And we're actually at a point now where we need to do a big proposal push again to take our research further, especially on the back of the results we've had most recently. So in the research space, what's exciting for us is the prototype vaccines we tested with Dr. Verwoerd. And for us, really the next steps would be how do we improve the efficacy of these? Do we add different adjuvants, for example, something to further boost their immunogenicity? Or do we vaccinate birds with different combinations of these vaccines? So there's still, we've only now touched the tip of the iceberg. We've shown a proof of concept. We need to open that door now a little bit further and see what's behind it. So, and then to better understand the immunogenicity elicited by these vaccine candidates. like I said, a big challenge for us is challenge material. And, you know, when we continue with this, what we'd have to do is we'd have to get live virus from birds that have been infected and have died. So we can harvest the virus from their organs and so, because that's our only source. Unfortunately, that is not necessarily reproducible, because lots of virus purify are going to have different and so. But at least to get to a point where we can say this is really promising and that it looks like we have efficacy on this. And then just some of the other aspects is, you know, together with this vaccine development component, we can also look at the testing, at the serology, at the PCR testing, where you track the birds you vaccinated, also to see what their profiles look like over time. That would give us really valuable insight also into what their immunogenicity profiles do as they proceed after vaccination. And education, making sure that all our breeders are educated in proper biosecurity measures, whether it be quarantining, cleaning the cages, what are we cleaning with, how are we handling, and just like due diligence from everybody's side.
Thank you very much. Alta, one last question, probably more directed at you. What are the possibilities of more international collaboration on these research projects and developing solutions?
Well, that would be our next call, is to sort of reach out, especially to the guys in Australia, because they're doing really, really relevant work, and, you know, to see if there is ways of setting up collaborations and sort of sharing the information, but doing something that is maybe a bit more Africa-centric for us, especially since, you know, Prof. Verwoerd mentioned, or Dr. Verwoerd mentioned, we're targeting African grey specifically, especially now in the startup phases. So sort of collaborating and sharing really valuable information. So we're not always starting from scratch, because researchers like to hide their information a little bit. And it's sort of to get the conversation going, because we're always going back to the beginning, and it's so unnecessary. And yeah, so it's building up those conversations and taking it further.
Thank you very much. Now, the second part of our way forward is, and everybody has pointed out, is that the importance of biosecurity and management. And I'm going to ask, I think, someone that especially now is doing his research on the use of disinfectants, Prof. Bragg, to maybe give us a bit of an input there, please.
Yeah, sure. Just remember, disinfectant isn't a disinfectant, isn't a disinfectant. There's a vast number of different products on the market, and some of them are good and some of them are not so good. You need to, whatever you're using, you've got to make sure it's effective for what you're using it for. As been mentioned before, this is a tricky little virus. It's a very small virus, and it's a naked virus. Naked viruses generally are extremely, extremely, extremely difficult to kill with disinfectants. Very, very few disinfectants are effective. As I also mentioned, because we can't grow the virus, you cannot claim that you've tested the virus against beak and feather. You can only claim that you've tested the virus, you've tested the disinfectant against closely related viruses. So what you need to look for as a producer is, has the disinfectant been tested against circovirus? That would be your first prize. Or has it, if it hasn't been tested against circovirus, has it been tested against a very difficult virus to inactivate, like parvovirus or adenovirus or reovirus? So you need to look at what the disinfectant has been tested against. I've seen many disinfectant labels where they have a long list of viruses. They're all envelope viruses. Envelope viruses are the easiest to kill of all. So just because it kills an envelope virus doesn't mean it's going to kill a naked virus. You can get disinfectants that are highly effective, but you have to look very carefully at what you're buying. The problem with disinfectants, as well as there's now becoming clear evidence that there's, well, with bacteria developing resistance to disinfectants. This isn't going to play a role with beak and feather. But if you're looking at a disinfectant to disinfect your flock against all sorts of other things, you remember there's a zoo of organisms that can be present in your flocks. So good biosecurity, good disinfection shouldn't only focus on beak and feather. You need a disinfectant that is going to be effective against all of the pathogens or potential pathogens that might be present. And bear in mind that there's growing concerns with resistance to disinfectants.
On that point, the question popped up from one of our participants was, what disinfectants do the labs use? In the research labs, and I suppose also in the testing laboratories.
When I was working with beak and feather and all the work that we've been doing with the resistance, we only make use of Viracula avium. It's proven efficacy. It's been tested against circovirus. It's been tested against the Gambora, the chicken anemia virus, adenovirus, reovirus, basically a whole range of naked viruses. So it's highly effective, and that's what we use and what we disinfect the lab with as well is Viracula avium.
Thanks very much, Prof. I'm going to go back to Christine. I unfortunately neglected to ask her comments on the way forward, but at the same time, we've added another little portion to your question over and above the way forward. What sort of procedures is applied in a laboratory to ensure the biosecurity and potential cross-contaminations?
Yeah, so we do extraction controls with our viruses or with our PCRs, and to ensure that there's no cross-contamination between samples. We also disinfect our labs every single day. We fumigate when we can fumigate. We sterilize everything in an autoclave, and obviously we try our utmost best to keep everything as clean as possible. We process samples separately. We do not process them together. And all of our machines are always disinfected. Yeah, so I think on the disinfectant part, there is always a chance of cross-contamination. That's not impossible, but I think all labs do our utmost best to not have any cross-contamination. I think it's also very important to have open communication with your clients, that if you think there is an issue, to communicate with them and say, listen here, I don't think this sample is viable anymore. Could you possibly give us a new sample? I know it's very irritating to the breeders, but we really believe in ethical testing and we really believe in open communication. Just on a side note as well, if we get like a low viral load, if there is a low viral load sample that we detect, it's probably not advisable to retest the same sample, like Dr. Dennis York showed you, that if you test it again, it might come out negative the next time, and then you're questioning results and you are unsure of what you have. I would say give it a week or so, retest your sample so that you can see if it's going up or down. And the way forward, I think biosecurity is very important. We've worked with a couple of breeders as well, where they started all positive and then they test negative after a while, after they've done all of their biosecurity, disinfected their samples and everything. So I just think go lab with the labs. Yes, the different labs get different results, but it's also dependent on the sample that they get. I think it's very important to be open.
Yeah. Thank you very much, Christine. Prof. Rob just wanted to make a comment.
Yeah, just to put a cat amongst the pigeons and make everybody nicely scared. I've done a lot of work in the poultry industry, and one of the big concerns is how can you evaluate if you've got rid of the viruses in poultry between flocks? You can do PCR, you take PCR samples from the floor, but guess what? They're going to come up positive. Even if the virus has been inactivated and killed, the DNA or RNA can still be present. So you need to make sure that you're killing the virus, but just bear in mind that the DNA can still be around and it'll still be picked up by PCR. even if the virus is dead. What happens in the poultry is there's a system where they put the chicks in, and then you take samples from the chicks at 7 days, and then you do real-time PCR to monitor for certain viruses, and then you can evaluate the efficacy of your disinfection program. But I just want to put out there that, even if you're using the best quality disinfectants, there can still be DNA that remains behind.
Thank you very much, Prof. That's very insightful. Are there any other panel members that would like to add a comment here on the biosecurity aspects?
Yeah,
I think
We've got Dirk coming up.
Yep.
Dirk, you can go first.
Okay, thanks a lot. Yeah, I just want to add, I'm a big proponent for the fogging route, for example, and fogging in the presence of chicks, for example, in chick rearing facilities, and etc. But remember that droplets, even the smallest droplet, do not penetrate, every crack and so on. So we've had experiences with this where, for example, there's residual contamination from one cycle to the next in the air conditioning system. So when you have an opportunity to do an all-in, all-out in the quarantine scenario that obviously is between cycles, you dismantle that entire air conditioning system, the whole thing, and that is washed and disinfected and so on, and then the entire facility goes through a fumigation stage. So gas do penetrate places where droplets do not. Obviously, it's the formaldehyde gas, and that's a very dangerous thing to work with. But if you can, and for example, incubator, chick rearing rooms and so on, where you have an all-in, all-out system, that would be the next step. Disinfection and cleaning is never a single step thing. It's always multiple steps. There's always the need for a soapy element to wash off the fatty layer, literally, and then the final disinfection. And in our case, we then go to a all-in, all-out system where there's fumigation, which is not possible in the presence of breeders and so on. So horses for courses. Thanks.
Thanks very much, Dirk. Daria, you wanted to add something?
Yeah, just on I think it is pertinent to mention this in the biosecurity section. I wouldn't advise, if the birds have tested positive in the past and you've run a certain cycle or whatever and it tests negative, to see those birds as negative. I'd highly caution against that, because now a lot of the breeders want to reintroduce and the infection flares up after a while or something happens. There's just something to mention there.
Thank you very much, Daria. I think that was very pertinent. I think we've concluded most of the basic stuff. And if you read between the lines and you take all the inputs, and I must say thank you very much to the panel for some really, really good inputs. There's a lot of information has come out. We're going to take a short break before we start with a couple of questions. We are unfortunately limited on the number we can do, but please, while the break is running, please submit your questions on the comment section. We'll handle a couple of them with our panel. Those that we don't get to, we will refer to our panel and we'll give you feedback later. But once again, thank you very much for everyone's participation. It's been very active, and I think we're moving forward, and the panel especially as well for their interactions. Thank you. right, thank you very much. We welcome our panel back. Now is our opportunity to answer us a couple of questions. The question will be put on the screen. I'll read it out, and then we will ask the panel members that are prepared to risk an answer to please put up the hands so I can see who I can approach, and we'll take it from there. Right, the first question is, other infections will create stress situation, making birds more susceptible to beak and feather infection. Anybody prepared to comment there? All right, Dirk.
Yeah, I think that goes without saying. We've alluded to that several times, but remember, it's a self-perpetuating cycle. The one opens the door for the other one, that builds up momentum, and then you get to a threshold where there's more of the first one, and so on and so forth. So different agents, you can almost say, feed upon the situation. So you'll have a beak and feather underlying break, so to speak, and then you'll have chicks dying of salmonella. And so on. So we see this in many hosts, in many species, in cattle particularly, that I work with every day. BVD virus is a immunosuppressive virus, but you don't see that as a break. They break with IBR, which is a herpes virus. So it is very true for every situation that one agent sort of preempts the situation for another agent. And this is where the diagnostic process have to roll that whole thing back to assist you.
I think it's very important to stress that the importance of good husbandry and good health management is key, as a lot of these outbreaks of diseases are multifaceted, to be clear. Right, the next question: what's the possibility that lab testing can become cheaper to get more breeders on board having their birds tested? Some of our lab people can maybe give us an insight into the costs. Christine?
Yeah, so every lab obviously likes to be competitive in the industry as well, but PCR also has a lot of reagents, and different labs use different extraction methods. So it depends on all of the costing of that for the lab specifically. I do think it's important that if you probably have a large number of samples that you're going to send out through the year, you're welcome to approach the lab and discuss discounts for you specifically. I do think that it is possible to come to an agreement to get more breeders on board to test. I know it's very expensive, especially if you're testing one bird at a time. But it's also important to remember that the lab also needs to kind of keep their business running as well.
Thank you very much.
I can also add after COVID, I mean, some of our reagents have gone up in price, I think 3 times. So it's difficult, or not 3 times, it's 3 times more expensive, which makes it tough on the labs. However, we are trying to stay competitive, and we have introduced a lot of things like the pooling of samples and just making sure that you testing on a regular basis instead of if you're pooling your samples and doing spot checks, it's a lot cheaper than actually after infection to now figure out where it is, and then you have to test your whole flock.
Thank you very much, Daria. Right. There's a very pertinent question. Our panel, what type of costing does the research require? Those that have actively Prof.
Hundreds of millions. Now, research is expensive. Like Alta said, you can't do research without good quality students. Like, for example, I was lucky enough to have Alta as one of my students, but it costs money. The students have to live, and then the reagents are expensive. So, there's a lot of money. It's very difficult to put a fixed sum on how much is needed. You need to look at a project. You need to do a proper project evaluation, and then you need to see what the costing is. So, but research is expensive. Unfortunately, it's not cheap. It costs a lot of money.
On that subject, can I ask how many projects are running at the moment directly related to the avian circoviruses and the feather disease?
From my side, I have one student on BFDV at the moment, and he's close to completion. So, which is why this next cycle needs to start. And like Prof. Bragg said, it's about sitting down and then deciding what exactly is it that we're planning to do, because we need to cost it when we're applying to our funders for money. And some of what we've done in the past is cheap, research speaking. It's never that cheap. But the immunological studies and working with the birds and all of that, it's quite intense. And students are sometimes slow. So ideally, you'd want a postdoc on a project, especially something that you want high throughput data for. So that increases that little stipend cost that you would build into your funding. So it really does depend on what you're doing.
Thank you very much. Daria?
Yeah, so just for the listeners, the cheapest cost would be the student, and I think a PhD student now is 180,000 Rand, so per year. 200. Okay, so from our side, we have one project running. We're just working with some of our clients to test the gathering of the virus to see whether it gathers in some of the glands, and to see whether that is a way of the virus just trying to disguise itself or just hiding away from the immune system. But that's a small project.
Thanks very much, Daria. Dr. York?
I just want to comment about pooling. Just be very cautious about it. We don't encourage it, not because we realize the price is expensive, but you run a risk. Each test is internally controlled for that one sample. So if you're pooling, we're not going to be able to control for all of the samples. Sometimes we've noticed that people do put spots of blood all over that piece of paper. So rather, we have a pooling process where you pay a pooling surcharge, where we do the extraction and we do the pooling. So just be very mindful. Again, it goes back to reducing any risk. In a negative environment, it's probably not a problem. But there's also other viruses that you're not sure are there. So just be very mindful. Don't just save on costs by pooling all the samples as many as you can. Rather, again, discuss that with the lab and be very careful about it.
Yeah,
thank you very much.
Sorry, we also only recommend it in the negative environments, not where you're trying to figure out which of the birds are positive, but just the spot checks on a regular basis to make sure that your cages stay clean.
Thank you very much, Daria. Can I have the next question, please? Right, the question is, PBFD more than with babies from raising babies via incubation, or babies pulled at 3 to 4 weeks of age from nests? Where would we find the most? Dirk?
Yeah, definitely from the nest. So if you say vertical transmission, which is a term often used, there are actually different pathways there. So there is the proper transaviarial transmission, which is, from my observations and what I understand, relatively small, a minor risk. The major risk is in the mother-to-child transmission. So if you leave your chicks for that first week or 2 or 3 or how long until you pull them, then there's a big chance of transmission to that chick. So that would be by far the most important one. And in a positive situation where you often are faced with a so-called healthy flock, but the chicks test positive and they die, one of the first things to do is to go to a egg pulling, clutch pulling model, and then rear chicks, from day one, incubate from day one. So you break that vertical transmission cycle. So that is by far the most important of those routes.
Thank you, Dirk. Just for our participants' interest, what is the incubation period of this virus? Dirk?
I really can't tell you. It depends absolutely on that interaction with the chick, because we've got this latency element. So the chicks will typically break not because of an incubation period related to the virus, but more to the stress bottleneck timing. So it would be, as I've said before, when they wean, when they feather, when feathers are halfway, you start seeing them to drop feathers. Those blood feathers are dropping, and that's often the first sign. And then they start dying. You open them up, and they've got acute viremia pathology, or they carry on for a while longer, and then they break. During or just with the transport and export process, and then they break 2 weeks later. But the infection actually took place a few weeks ago. So it's not like many of the other viruses that we work with that we say, well, incubation is 5 to 7 days, and that's the classic sort of model. It is completely different, and it is more related to other latent viruses like herpes viruses and so on, where it's a big part of that whole pathological pathway.
Thank you very much, Dirk. Just a follow-up question to Daria. In connection with the testing, what is the youngest age a person should test, or the ideal age a person should test a chick if you want to determine the status of beak and feather in a nest from a breeding pair?
Yeah, I think there hasn't been tests done on the earliest you can do it, but I think if you give it between 1 and 2 weeks after birth, it would be a good time to test, because the younger they are, the lower their immune system, so the more susceptible they are to various infections.
Thank you very much. We have time for another 2 questions. Do we have areas in South Africa where the virus is more prominent, was the one question. Any of our researchers or labs been able to identify a pattern there?
Yeah, it's more prevalent where the most parrots are.
It's a good answer.
All our other participants are shaking their heads. Can we have we have a next question. It's in Afrikaans, I'll translate it. For the labs, what method of submission of samples would they prefer? Must we freeze and then send, or that we rather send a fresh sample? And this is probably more in the case of a dead bird or material. Would freezing have any influence on testing procedures, basically?
I think because we're using the FTA cards, I don't think it would be advisable to freeze the FTA card, or do they mean actually sending physical samples?
It seems like they want to send a physical sample, yeah. So it'd be organs.
Like a dead bird.
Like a dead bird.
Yeah, we don't recommend sending dead birds at all. We try not to handle birds in general, because it increases the risk of cross-contamination in the lab with all the additional feather dust and those kinds of things. So rather have your veterinarian draw blood and send that to us, or taking a proper sample. It is a more difficult thing to work with, because you have additional extraction methods that you have to take into account, but you can a normal blood sample would be better.
Thank you very much, Daria.
Yeah.
Continue.
No, I don't know if any of the other labs have anything to add there.
Doesn't seem like that. Dirk wanted to make a comment.
No, just a practical issue. I find it very practical, even in a frozen bird where you can't really make out much in terms of macro lesions, etc. At least you can see that there's a massive spleen. The spleen is a very good organ for any viral extractions in acute viremia. So it's easy to take out the spleen, cut it in half, and use that as to swab the paper and then send the paper in. So that would be a good way around that. If you want a proper macroscopical evaluation of the pathology, do not freeze. Send it to your veterinarian as soon as possible. Take pictures if you can't. There's many guidelines and so on. I have many people that I've trained how to cut it open, send me the pictures, and then at least you get a broad idea of what's going on. But that would be just a few practical suggestions.
Thank you very much. We have time for us. I see Dr. York wants to make a comment.
No, I was just going to support that. An imprint of tissue sample on that paper works very well. I also say, please don't send us the bird.
Thank you very much. The last question for today: what impact does finches and canaries have, and can they be carriers? All right, Prof. Bragg.
There are some reports of canary circoviruses. How it's related to beak and feather, I don't think it's very closely related, but there are various other circoviruses in other bird species. Like, for example, racing pigeons. Racing pigeons have a big problem with young bird disease, which is the circovirus.
And I think a closely related one would also be in the case of budgies with French molt.
French molt, yeah, absolutely. Yeah.
Okay, thank you very much. We just have a comment or two from people, and please, all the participants, if you found the seminar insightful, please add a comment, post it, so that we can get good feedback, so that we know what our approach is going to be forward. I think you can all read the post there. We have some more posts there. We're going to share a couple of them. While we're busy with that, I would like to thank our panelists for their very, very valuable inputs. Having a bit of a background, you can pick up a lot of information in between the lines, and I think that was very insightful. And I think the important thing is to build on what we've discussed today and map a way forward to try and resolve a disease that's been with us for quite a while and has challenged many scientists and still will, because it's a very, very intricate little virus. But we appreciate that what you have done collectively, and probably hopefully still will do, to try and resolve this disease for us. And from our side, from the Parrot Breeders Association, you have our support, and if you need additional support or inputs, please be free to approach us. And we thank you for your participation. Prof. Bragg.
Yeah, before we end off the thing, Anton, I think it would be amiss of us not to thank you for the way you've handled the session. You've done an exceptional job of keeping everybody on track and keeping the questions coming, and you really did an exceptional job doing this. Thank you very much. Much appreciated.
Thank you very much, Prof. We appreciate it. We close down. The Prof. Dr. York, you want something? Okay, well, I'm going to open the floor to the panelists for a last comment each, and then we'll close down. Thank you. We'll start, Dr. York.
No, I was also going to say thank you to Guy as well for putting this together. He was up all hours of the night last night and before. But I think maybe Dr. Pansel, if you do get some viral material, and if you are able to make it into a reference sample, putting a panel together and then circulating it, I think it'll be an excellent idea, and I think we will be prepared to pay for it. So that's one way of getting a little bit of money. But thank you for this most interesting presentation.
Dirk?
Yeah, just a sincere thank you to all concerned in behind the scenes and arranging. Apologies for the technical glitches on my side, but I also found it very helpful and insightful. And I think going forward, we need to look at this not as individuals or individual groups. It really has to be a cooperative engagement between a lot of people. It's only through teamwork that we will really progress and finally get to the stage where we all want to be. So we're in an excited phase. I think we're in between a phase and we're at the verge of breaking through in the next phase. Money is needed, there's no question, but we also need material. So if there are outbreaks, please contact us. We do need material. We can eventually get to a stage of circulating standard material and so on. And that is essential in moving forward. Thank you very much.
Thank you very much, Dirk. Christine?
Yeah, I would like to thank you for letting me be part of it, and also getting the lab side of it, so that people could understand the lab testing a little bit better. And it's not one lab against the others. Obviously, we are situated in different areas, and we try our best to satisfy our customers to the best of our abilities. So it's not us against Lumigen or us against MDS. I think we all kind of work together to help out the industry. So I think it's very important that we do have these webinars so that people can understand it better. So thank you so much for inviting us. It's been a real pleasure, and I've also learned a lot here from it.
Thanks very much, Christine. Daria?
I think it's now been 9 years that I've been working with PASA, and just the work that they've been doing just for the industry is excellent. And I'd encourage everyone to be part of what they're doing, registration of birds, assisting with all those difficult things, and just the awareness for the industry. They have really been doing great work.
Thank you very much, Dr. Alta.
Yeah, thanks for inviting me here today. I think it's great to have all of us in the same conversation, because it's the first conversation of what I hope to be many more conversations. Because as I said before, this is not something we're going to solve or address by ourselves. We need to work together in everything from doing the research and the testing to doing the grant applications and getting different experts into those applications, so that people want to give us the money and trust us with their money to do their research and to take this further. I'm very excited about where we are with what the research is showing at the moment, and hopefully for the next one we've got some more results to show.
Thank you very much to everyone. I'm going to end off by thanking PASA. It started off with a bit of brainstorming and a couple of ideas, but we've put together a team that made this a reality. We challenged each other, we set goals for each other, and I think I can confidently say that the team, I'm not going to mention names because I will definitely forget someone, but we challenged each other to achieve this, and I think by the end of today we've achieved our goal. And please, as I mentioned in the beginning, this is just the first on beak and feather disease. There will definitely be some more, and some more very interesting webinars that will be following this year. Please support your organization, get involved, and we can just grow to greater heights together. Thank you very much, and enjoy the rest of your Saturday.
Thank you.
Thank you.